I hope everyone can hear me. Welcome back to the workshop, and I think I'm the last talk of the day, so thanks for hanging around. And yeah, if everyone's ready, I'm going to talk a little bit about BigBrain Warp. So yeah, for those who don't know me already, I think I've been introduced a little bit. I'm Casey Paquola, and I'm leading a new multi-scale neuroanatomy group in INM1. At the Forschung Center in Jülich. But for the past 3 years, I was a postdoc at the MRI with Boris Bernhardt. And together we've been developing a toolbox for multimodal integration of big brain called BigBrain Warp. In the first half of the talk, I'd like to discuss the conceptual backgrounds for this and the motivations for building the toolbox. And then I'll describe 2 examples of how we've used integrated big brain MRI analyses in the past. And then in the second half, we'll delve into the toolbox itself and finish up with a little demonstration of how you can use it. Feel free to put your hand up if you have questions or just ask in the chat or anything. Okay. Here we go. Okay, so in vivo neuroimaging has allowed, I think, really comprehensive characterization of the macro scale landscape of the human brain. For example, functional MRI shown here enables visualization and analysis of ongoing fluctuations in cortical activity. I think this video really epitomizes how we strike this balance in vivo imaging between spatial and temporal resolution. And this has profoundly changed our understanding of the dynamics of cortical function that occur from second to second, but also across a lifespan or even across evolution. And this stems from major advantages of fMRI, that we can roll it out in a large cohorts of individuals. And then we can also assess similarities and differences. And this has also enabled us to examine the coordinated functions across the brain. So inspecting this functional connectivity, as it's usually called, has led to new depictions of cortical organization. The neuroimaging field has embraced some of these conceptions, such as clustering the cortex into communities and parcels, or this principal gradient of functional differentiation. And these canonical examples demonstrate how complex patterns of cortical activity can be deconstructed. And then they become this useful framework for us to understand other variations across the cortical mantles, such as neurobiological properties or inter-individual differences. And then on the other side, we have big brain, which is really helping us to uncover regional variations in the composition of cells. And it really has to have this flexible machinery so we can relate cytoarchitecture to in vivo imaging. And I think this is especially interesting because the relationship between cytoarchitecture and function is very complicated and unresolved theoretically. There have been a couple of linking principles that were primarily identified or theorized in the 1990s. These relate to there being common axes of cytoarchitectural and functional differentiation, and that you could predict connectivity from cytoarchitectural similarity. But these were primarily developed in animals, non-human primates. And whole cortex analyses of these principles in humans have been really lacking because it's really hard to have these combined data sets where we can look at cytoarchitecture and function. So I'd like to start off by discussing how our integrative big brain MRI analyses have helped us to test these principles in the human brain. So let's start with the first one, which comes mostly from Marcel Mesotemporal. So he described cortical organization as a series of rings, the outermost of which contains primary sensory areas, so you see V1, A1, and then each ring is a synaptic step inwards, so you get deeper and further away from this external information. You also see that cytoarchitecture varies systematically across the rings. So the outermost rings have very high laminar differentiation. You see 6 or more layers, and this laminar differentiation decreases as you move inwards. Then combining this with primate electrophysiology, Marcel Mesotemporal noted that also the type of function changes as you move in the rings. So you start at very high dimensional sensory processing. It moves to unimodal, then heteromodal, and then even amodal as you move into more paralimbic and limbic areas. So it seems the type of functions are becoming more abstract and also more transmodal. Mesotemporal proposed that these are really systematic changes that are concordant in some ways, and these are the most dominant changes or axes of differentiation in the primate brain. And he labeled this as sensory fugal axis and suggested that it's common cytoarchitectural and functional axis. So we sought to test out this principle using BigBrain a few years ago. If you want more details on this work, you can find it in a 2018 paper that we wrote, but let me talk a little bit about specifically how we tested that principle. So we first of all tried to characterize cytoarchitecture across BigBrain using staining intensity profiles. So these here, they reflect the cell staining intensity as you move across cortical depth, so from the pia to the white matter. So here you can see the different colored lines are two different areas of the brain. You see they have different staining intensity profiles, which means they have variations in the depth-specific cellular density and soma size. So then we can take these profiles across all of the big brain and correlate them with each other, making this cytoarchitectural similarity matrix. And then we applied diffusion map embedding to this matrix, which is a type of nonlinear dimensionality reduction. So it's going to find the principal axes of variation in this matrix. We find two really pop out as explaining a significant amount of variance in that matrix. So if we look at these, the first is this anterior-posterior axis. So each of these is an eigenvector, each of these dots. So you can think of it similar to a PCA, but diffusion map embedding has a better balance of global to local kind of geometries in its algorithms. So here we have a loading of an eigenvector. So this is the first eigenvector, and you see that it's negatively weighted in posterior areas and positively weighted in anterior. So we tend to call this an anterior-posterior gradient of cytoarchitectural differentiation. And then the second gradient here looks to transition from more sensory areas, but then it moves through to the opposite end of the eigenvector is these parahippocampal areas and these orbital frontal areas as well, which is more similar to what we would expect based on the maps produced by Marcel Meslem and others about laminar differentiation. So this looks more similar to what we would assume a sensory fugal gradient looks like. So these are projected onto the big brain surface. But then what we can do is transform these onto a standard neuroimaging template like FS average. And that's what BigBrain Warp really helps us do. It's a lot about doing these transformations. So here we see this histological gradients projected onto FS average, which most people in neuroimaging have used at some point in time. And that's also a standard surface that we can use for in vivo imaging. So we applied a similar technique using microstructural sensitive MRI, specifically our quantitative T1, also works with T1 over T2 or magnetization transfer, but applied a really similar technique to what I just displayed, where we take these profiles, like depth-wise profiles of changes in intensity, we correlate them, we apply diffusion map embedding, and we see what the principal axes of variation are. And we see here, this principal axis is really clearly transitioning from these sensory areas in blue towards these more limbic areas in red. And then we also compare this to functional gradient. This was really popularized by Daniel Margulies and others, where we look at the functional connectivity matrix across the entire brain and apply diffusion map embedding. And you see this transitions from sensory areas in more blue to the default mode in more red. So if these are our principal axes of differentiation, then based on Marcel Meslem's theories, we would expect there's a high level of collinearity between these principal axes. And what we find is this anterior-posterior axis is not so related to the others, but we find a very similar correlation. So here it's the, you see the row values here, it's a Spearman correlation, and the size of the circle is just trying to give you a quicker view of this as well, of the strength of the correlation. But you see that this second cytoarchitectural axis is closely aligned to the microstructural axis, so from in vivo imaging. So it's a 0.64 correlation, and it has a 0.52 correlation with the functional axis too. So it seems to be some collinearity that aligns with Marcel Meslem's theory that cytoarchitectural and functional differentiation are aligned across the human cortex, at least the most principal axes of cytoarchitectural and functional differentiation. Okay, so then on to another principle that we can test. This comes from mostly Helen Barbas's group. They introduced the concept of the structural model in that microstructural similarity can predict connectivity. They showed that in cats and in macaques, that the more similar two regions are in their lamination, the more connections they share. Then using neuronal density as a proxy for cytoarchitectural type, Boole and Hilgertag clearly demonstrate this relationship in the macaque cortex. So here they are trying to predict projections in the macaque cortex using different features, either the similarity in neuronal density, the distance, or similarity in thickness. And you see, just taking the similarity in cellular density and the distance between two regions, you can have quite high level of prediction of the existence of a projection. Then the structural model also makes claims on how the structural type predicts the laminar origin of projections. So here you have dissimilar cortical areas. So they have different types of cytoarchitecture, like an agranular, where there's not clear lamina, versus eulaminate 2, where you can see very clear layers here. Now they project to very different layers of the cortex when they're going between each other. Whereas if you look at between disc granular and eulaminate 1 that have more similar cytoarchitecture, they're projecting to more similar layers. The connections kind of go to and from more similar spots within the depths. And this is also really interesting because this can be a marker for feed-forward, feedback processing. So feedback processing going from agranular to eulaminate 2, so more fugal to sensory. See, the origin is much deeper, whereas it's the reverse if it's feed forward, where you're going from eulaminate 2, which is more sensory, deeper into the brain as well. And so we can see these relationships here between differences in cytoarchitecture and the direction of connectivity as well. So we were quite interested in exploring these ideas within the mesotemporal lobe specifically, because that's where we see such profound variation in cytoarchitecture, because it has both allocortex, which is agranular, so you really don't see 6 layers, at most you'd maybe see 3 layers, and it's also abutted against this isocortex, which has clearly 6 layers. And there've been previous research showing that there seemed to be a gradient of change from isocortex, so out here in kind of the parahippocampal area, and then transitions through to this hippocampus that's allocortical and slowly seems to lose some of those layers and become more agranular. So we took this isocortical model of big brain, so you can see that in purple there. And we also combined that with a manually segmented version of the hippocampus that comes from Jordan de Cracker and Ali Khan at Western. So here you see this in orange. And one of the very cool things about this hippocampal model is that they also produced internal axes within the hippocampus that tell you which way is the inner and outer part of the hippocampus. So that aligns with the pia and white matter boundaries of the isocortex, and an axis for proximal to the entorhinal cortex or distal. So distal would be deeper in the hippocampus, more dentate gyrus. So we created a little nifty way to slice off this part of isocortex that skips over the hippocampus and instead link the isocortex to the hippocampus here. So you have this continuous model of the mesotemporal lobe. That's what you see here. Then we took at the intersection, we just looked at the geodesic distance along that surface mesh. So we have a, this geometric measure of I-star allocortex. So it really follows the curvature of the hippocampus. So from parahippocampus, entorhinal, kind of going all the way in, it goes through all the subregions and then hits the dentate gyrus. So now we had this really nice new continuous surface model. So we could sample these staining intensity profiles in a continuous manner across the entire mesotemporal lobe. That's what you see here. And you can see quite obviously it jumps out that there's systematic differences in the intensity and placement of high staining intensity as you move from more isocortex to allocortex. And also, if you want more details on this, check out, there's an eLife paper from 2020 where we talk more about the cytoarchitectural changes, but I just want to touch more on the functional model here. So here we then projected from the surface model into big brain space, and then we used a nonlinear transformation to go from big brain volume to ICBM 152. So we're now in a standard space that we could use for functional neuroimaging. So in one analysis, we used this to define seed regions and calculate the effective connectivity within the mesotemporal lobe. So here we have a set of seed regions going from isocortex into allocortex. And then we calculate effective connectivity using a dynamic causal model. So you see that matrix here. And then we could test whether the effective connectivity is related to the proximity along this axis. So whether the effective connectivity is stronger along neighboring regions, or the effective connectivity seems to show that there's a lot of jumps across the axis. So is information flowing along this, this eye-stylocortical axis? And that is indeed what we find, actually, that effective connectivity decreases if you have higher deviation from the axis. So stronger effective connectivity if you're going to neighboring regions. So there seems to be a bit of a flow along this axis. And we find here you have each direction. So if it's going into the hippocampus or back out, there's still that strong correlation. So it didn't seem to preference feed forward or feedback. It was just both ways. There was a strong evidence that information is flowing along this axis. And then finally, in another analysis, we sought to expand this scope to large-scale cortical dynamics. We tested whether functional connectivity between isocortical areas of the rest of the cortex with the mesotemporal lobe was associated to the eye-stylocortical axis. So we find that there are some regions where this is the case. The red regions, for example, they have stronger connectivity to the allocortex, towards the hippocampus, whereas these blue regions have stronger functional connectivity to these more isocortical regions. So it seems that within the mesotemporal lobe, we already know there's a lot of evidence about this anterior-posterior axis that is related to the functional connectivity to the rest of the brain. But now that we have this histologically based model of the eye-stylocortical axis, we could also find that there's an intersecting axis that also organizes functional connectivity. I think that's been really largely overlooked in the past because it's been so hard to model it just based on in vivo imaging. And that's the real advantage of then integrating these super high resolution histological models into our in vivo imaging pipelines. So I've shown some examples of us being able to test and expand upon established linking principles between cytoarchitecture and function using these integrative big brain MRI analyses. But I think that's just the beginning of what we can uncover. We could apply similar types of workflows to interrogate principles from other work as well. So for example, work by Alex Goulas has shown in the macaque that prediction of connectivity from cytoarchitecture varies depending on the distance and the length of the track. So this is something that we could definitely expand upon using big brain and MRI in conjunction. Then that paper, as well as work by Garcia-Surfacebased et al. In the human frontal lobe, highlight that there are many cytoarchitectural features that vary across gradients. So it's not just laminar differentiation or these principal axes, but also the size and morphology of neurons. So all of this could be more comprehensively characterized and related to macroscale features using big brain. And then finally, microcircuit models are primarily based on primary visual cortex or other isocortex, but the microcircuit could look very different in agranular cortex. So here you see a theory proposed by Bull and Hilgertag as well, that the agranular type microcircuit could look very different to this highly structured, high laminar type of microcircuit. So I think there's real promise in using big brain and MRI analyses to incorporate cytoarchitectural information to improve computational models of large-scale brain function. So that's where BigBrain Warp comes in. We've created this toolbox for integrative cytoarchitectural analysis. So on one side, we aim to facilitate the characterization of cytoarchitecture. And on the other side, we want to bring this together with a real wide range of different types of in vivo imaging. So here we have what we aim to do with BigBrain Warp. So in the purple is really what is contained in BigBrain Warp. So if you start off with, in this example, big brain on one side and MRI on the other, in both cases, you need to do some type of pre-processing to extract some features. With BigBrain Warp, we've already produced a lot of features that might be helpful. I'll talk about a little bit more in a second. Then you need to do these transformations to bring them into a common space. Also in BigBrain Warp, we have some really simple functions I'll show later of how to do this. And then finally, you need to have very smart kind of workflows that take into account the advantages and drawbacks of both sides of this picture. And so we've created a set of tutorials that are on the BigBrain Warp website, and they're also in a paper that we've just published on BigBrain Warp as well, to help people learn from what we've been learning from the past few years in doing this as well. So in terms of the cytoarchitectural features, now I'm just going to turn on my light for 1 second. I realize I'm quite dark. Hi again. Okay, so the cytoarchitectural features. So one effective approach that I've already mentioned is that we can characterize this cellular architecture using staining intensity profiles. So here, you take this histological section. This was really pioneered by Karzillis and Axel Schleicher in 2D, and we're using it in 3D here. So you go from the pier to the white matter, and you just sample the staining intensities along this line. And here you end up with a nice profile here. Even though with BigBrain at the moment, this is at 40 micron or 100 microns in terms of volumes. So we're not at a cellular resolution, but we can still see certain cytoarchitectural features jump out at us here. And this is even an average profile. So we're not at the super high resolution, but you can straight away see that there's this layer 4 peak that we would expect from changes in cellular density as well. And you see this large increase as you move from layer 1 to layer 2. So it's sensitive to different types of cytoarchitecture, even if we're not perfectly at this cellular resolution. And then we also have contained in BigBrain Warp these cytoarchitectonic models, so such as the cytoarchitectural gradients I showed before. We're also incorporating these laminar definitions that Conrad Wagstill has made, as well as morphometric models, so such as the hippocampus I showed before, and the then mesotemporal confluence, which is the conjunction of the isocortex with the hippocampus. And then at the heart of these BigBrain MRI analyses are the transformations between BigBrain and standard neuroimaging spaces. So we have BigBrain and we have BigBrain Sim. I'm not sure if this may have been gone over already, but BigBrain Sim is a nonlinear registration of BigBrain to ICBM 152. It's not exactly the same as ICBM, you can obviously see. So there's also another step that we can do to possibly optimize that. But we have these volumes, and then we also have their corresponding surfaces. And then we have standard surface templates like FS average, which also covers FSLR as well, which is in HCP Workbench packages. And really the aim of BigBrain Warp was to pull together a lot of the previously established types of transformations in the field and amongst big brain researchers. So this transformation from BigBrain to BigBrain Sim was done by Claude Lepage, like at the very kind of start of BigBrain. And then also this optimized version by Zhao et al. For better registration to ICBM 152 in volumetric spaces, and really amazing work by Lindsay Lewis on surface-based registrations from BigBrain to FS average and FSLR as well. So the aim is to kind of put all of these under the hood of BigBrain Warp, so we can keep updating these as the field progresses. And all that the users need to know is that you put in what you want to put in and you get out, what you want to get out really. So this is all covered under the hood of the BigBrain Warp function. There's a range of different transformations that it calls. So you can basically think of these as 8 different transformations that are involved, going from volume to volume or volume to surface. And these are all the different forms that it can take. But that will maybe make more sense when at the end in the demonstration, I just show you how the different parameters that you put into the function. But safe to say you can put in whatever data you like in this diagram, and you can ask for whatever type of data you like back from this diagram as well. So there are 2 principal methods of transformations that are captured within this that I've touched on. So the first are the volumetric types. So these involve concatenated linear and nonlinear registrations based on image contrast. So there's this original BigBrain to BigBrain Sim one. And then there's also the Zhao et al. Approach, which use an inverted contrast of an image as well as an intermediary atlas. So when considering these approaches, it can be useful to consider how the transformation distorts the original image. So we can look at the Jacobian deformation fields for this. So here I've just taken a few different lobes of the brain. So you could see how this could also work for a region of interest. Maybe you have a region of interest and you want to know whether the intensities are really expanded or contracted within that region of interest specifically. So that's why it can be really helpful to look at the Jacobian deformation fields. And we can also compare how different approaches impact the expansion or contraction of the image intensities as well. But we also want to evaluate the accuracy of our transformations, of course. So one way to look at this is the alignment of manually segmented regions. So we take labels of subcortical regions that were manually segmented on BigBrain or ICBM 152. Then we apply the transformation to the BigBrain labels and evaluate how well they overlap with the native ICBM labels. So here you can see it with BigBrain Sim, and here's with this optimized approach by Zhao et al. And you can use the dice coefficient to look at the regional overlap. And here you can see this is really where there's a big improvement of the Zhao et al. Procedure. It actually uses shape priors of these subcortical regions as well. So you can see it's really boosted in that respect. And that's something important to keep in mind depending on what type of analysis you're doing as well. But then there's another way, a more traditional way to determine accuracy is by looking at the anatomical fiducials. So in a similar manner to the regions, you take these landmarks that are manually defined on ICBM and BigBrain. Then the landmarks are transformed from native BigBrain to ICBM using the transformation that you provide. And then you calculate the distance between the native and transformed landmarks. And so here we actually see that BigBrain Sim and Zhao et al. Are not so dissimilar in the accuracy. But overall, I think it's really nice to recognize that different regions might have better accuracy than others, depending on what procedure you use. So it is quite a handy thing to integrate into your analyses. And it also provides a nice estimate of the degree of uncertainty that we should have in our analyses. So here, the degree of uncertainty is a maximum of about 7.5 if we use these anatomical fiducials in millimeters. So this is something good to keep in mind, so we're not making over exaggerations of how precise our analyses really are. And then the other form of transformation is surface-based. Once again, we can consider the degree of deformation invoked by the transformations by looking at the surface mesh. So here we have 3 different types of deformations of the surface mesh that we could inspect. And then we can use predefined landmarks on the native BigBrain and on native FS average surfaces. So once again, we transform the BigBrain landmarks. We have these ones, we transform them onto FS average, and then we look at the distance between the native FS average ones and the ones transformed from BigBrain. And here this helps to show the advantage of an optimized multi-modal surface matching algorithm that was developed by Lindsay Lewis. So from using just sulcal depth, you have this wide range of errors or misregistration. But when she has then combined this with curvature information as well as optimizing the input mesh, you see a real minimization and reduction in the degree of error here, as this can provide also this good barometer of the degree of uncertainty in different registrations. And as the field progresses, it's something that we can always check and might be helpful depending on what region of interest you're looking at. So overall, BigBrain Warp is designed in a really modular way, and it's made to continue being developed over time as well. So we know that certain transformations might work better in certain situations. We want this ecosystem to continue growing, and that depends on your input as well. So if you'd like to join the team, please just get in touch. We're more than happy to have more collaborators. And if you've conducted research on BigBrain that you'd like to share, we'd be happy to incorporate it into the repository or direct BigBrain Warp users to it as well. And also the current infrastructure is primarily based on our needs as well. So if there's features or options you'd like to see be added, feel free to get in touch, and we can do our best to see if we can make that a reality. And you'll find a lot of information on BigBrain Warp in the Read the Docs as well. I'll touch on a few things in a second, but just before we get into the interactive section, I just want to acknowledge and thank everyone who's been involved so far, especially Boris Bernhardt, my mentor from the MRI, who spearheaded this project with me, as well as this has been a great collaboration between Alan Evans' team at MRI and Tim or Catherine, who I'm working with now at INM1, and the external collaborators who have helped along the way, especially Tristan and Ali. So thanks to everyone who's been involved. And I can just give a little bit of background on how to get started and just to run a couple of scripts, but it's a pretty simple toolbox. This won't take so long. And yeah, feel free to just jump in and ask questions at any time, but otherwise I can just take a few minutes to give a brief overview of how to install and things like that. So I think the easiest way at the moment is using the Git clone. The dependencies now are only Mink2 and a few Python libraries. We've really purged a lot of other libraries recently, and Mink2 is not so hard to install anymore. And the reason I'm also recommending the GitHub is because we keep it constantly updated, whereas the Docker is going to be every so often updated. So if you want the newest releases, try the GitHub version. Otherwise, yeah, every so often we'll create a more stable release of the Docker and we'll advertise on the website when that's ready as well. And then, oh, my slides have just frozen. Here we go. Okay, and so there's one main function in BigBrain Warp just called BigBrain Warp. There's 4 required parameters. These are the input space, the output space, your working directory, and a descriptor. All the naming conventions in BigBrain Warp are based on template flow. So you don't necessarily choose the output name, but it's determined based on your working directory. That's the directory you'll output to. And then this descriptor. So it will tell you which template it's ended up on, which hemisphere it belongs to, if that's relevant. And then you use this descriptor to give it some notable name as well. And then you can input either volumetric data. So this is assumed to be a whole brain volume. It doesn't have to be fully populated, but it needs to be a full whole brain volume aligned to one of the volumetric spaces. That could be BigBrain, BigBrain Sim, or ICBM. And then otherwise you can use surfaced input, which you provide each hemisphere separately. There's a range of formats that we accept, different types of GIFTs, ANOT files, curve files, text files, and this can be on FS average or FSLR. And then there's a few optional things that you can also consider for interpolation. We have a lot of defaults already built in, but generally it's going to be either linear or nearest neighbor. For the volumetric, there's a wider range. We use Mink form of transformation. So we essentially have each of the interpolation methods offered by Mink in these options, sorry. And then you can choose the output type. So that means that you can go from a surface to a volume if you specify it. Once again, we build in some simple defaults, but it's also handy to just put in, I want to go from you give in volumetric data, but you can say out type is actually a surface. There's some limitations in that though. We don't have it made so you can go to the FS average volume. If that's something people really require, let me know. But we kind of just went with what makes the most sense based on most typical pipelines as well. And then you can also specify the output resolution for volumetric data. It's going to default to 1 millimeter, but you can change that to whatever you like actually. If you go super high resolution, just be warned, it might take a while to run. And also for the output density of a surface mesh. So this is especially for FSLR because there's a 164K mesh and a 32K mesh. So if you're using FSLR, feel free to specify because I think a lot of people actually use the 32K mesh. Okay, so how does this run? They say once you install it, every time I start running BigBrain Warp, I just run these 3 lines of code. I move into my GitHub repository. I pull it to make sure I'm up to date. And then I just source this initialization script to make sure it makes sure that everything is there and just sets up the environment a little bit. Then I define my working directory and I run BigBrain Warp. So here you have an input, which is BigBrain. And the input volume here is this isoalicortical axis, this MTL axis, which is in BigBrain's space. And then we're going to use an interpolation. I chose nearest here for some reason. And then you're defining the out space, which here it's going to be ICBM because we want it to be in another volume space so we could do functional connectivity from, for example. to find the output resolution. And then we have a little tag with the descriptor, could be, I think that should be MTL axis, and then the working directory. And you can see this is going along this path. So it's a volumetric input. It's BigBrain template that we're starting off with, and we want to go to another volume. So this is just one of the forms of the transformations that's housed within BigBrain Warp, but all of this should be handled underneath the hood. So you just have to tell it what you want. Okay, so maybe what I can do is actually just show you what that looks like, and anyone can ask questions as well. As I just show you how that it looks quite nice when I run it. Okay, let's see. But yeah, feel free to ask questions if there are any. Okay, so here I'm also just viewing with Workbench View, which is HCP style thing. And so here you have, this is BigBrain Sim. Here I've just loaded the 400 micron version so that my computer doesn't overload. And here you have this isosceles cortical axis of the hippocampus. And then we've transformed it to ICBM, and it should look quite a little bit better. It should be a little bit better aligned because it's just going from BigBrain Sim to ICBM. But you can see that this is a histologically defined axis now on ICBM, and that's what we use for that project I just showed before. And you can even see that it has this good resolution. So you have this deepest part in here, and it kind of transitions all the way out. So that's one such example. And I can show another one. If we just go back to, yeah. So another example would be going from BigBrain surface to FS average. Really, the only main difference here is that you're going to put in the data on left and right hemisphere. And instead of defining the output resolution, here I've defined the output density. So we're going to have it on a 32K density mesh. But generally, I think it should be relatively simple. And there's example, this code that I'm presenting is already contained in the GitHub. You have all 8 forms of the different transformations that are possible in that script as well. So hopefully that answers a lot of questions of how to use this toolbox, but feel free to get in touch if there's any more.
Maybe it's a bit of an ignorant question. Maybe first also like thank you for this like great talk. It was really insightful and also really amazing to me like what you did. And yeah, and Katja and Roberto agree, and rightfully so. Like I'm really impressed. So I was just wondering about is this at all in your mind like translatable to the cerebellum in some way? So what I want to really start doing in the PhD, start looking at the microstructural functional link in the cerebellum, but then of course there's different different problems there. How do you look at this issue?
Yeah, so I definitely think that you could, similar to what we tried to do with the mesotemporal lobe, is use some type of manual segmentation of the cerebellum that might be too hard to acquire in vivo imaging. And then the nonlinear transformations I showed have not been evaluated for the cerebellum. So I wonder if it'd definitely be helpful, I think, to try to just manually segment some larger regions that you could identify both in vivo imaging and histology to make sure that there's a good alignment of the registration from BigBrain. But I think that if you're looking for to register some finer details, and especially axes as well, I think that was quite interesting, that maybe not just going for a tiny region, but actually going for some histological axis of the cerebellum and then transforming that, or some type of cytoarchitectural feature that you see in the cerebellum. I think that could definitely work. The only thing I would be wary about is just double checking that the nonlinear registrations work well in the cerebellum, because they haven't been evaluated in the cerebellum as far as I know.
Right. So this would then, because I'm now reading up on the work of Wachtel as well.
Yeah.
So this kind of like validation of that the big brain actually matches up. For example, they do it for like cortical thickness there, but that it matches up with the histological atlases of von Economo and such. So those kind of like things you would like also do in the cerebellum, and just start from a manual kind of like yeah, some kind of like cytoarchitectural difference that you find interesting. And
yeah, I think that'd be really fascinating.
Okay.
Yeah.
Okay, thanks. Yeah,
that's really cool. Yeah,
I
I think that.
Sorry, yeah, I'll have to wrap my brain around it, like no pun intended there, but yeah.
Oh, that's good.
Yeah, thanks.
Okay, thanks. Cool. If there's no one else, I'm happy to pass it back to Susanna to wrap up, or I can just say, see you all tomorrow. Oh, I think, okay, there's a question actually. Let me read it out. So it says, from your talk and your papers, it seems that most common to transform big brain data into a template space. Is that correct? Curious what you think the pros and cons are of instead taking template-based data, e.g. FS average, onto the big brain for anatomical functional analyses. Yeah, we do tend to go from big brain to imaging spaces. One advantage of that has been that we tend to do one very detailed cytoarchitectural analysis on big brain, and then we transform to FS average and do a lot of different analyses on there. And also, if we transform it to FS average, for example, we can then transform to individual subjects from there. So that has just been a little bit more practical for us, if anything. I think that one thing to consider is always the changes in resolution that you're going to be putting the data through as well. So it's going to be a little bit dependent on the analysis that you're doing. So I think, yeah, there's a pragmatic concern of just how many transformations do you want to do. It can sometimes just be easier to do one to FS average, and then you have an easy way to go from there. And then also the changes in resolution to make sure that you're not overestimating the precision as well. So for example, oh, and I think Lindsay actually had a small comment on this one as well. And I think she should be a very good person to answer this.
Okay, I didn't want to interrupt you. Are you sure you're
No,
done?
no, you're great. Yeah, go for it.
Okay, it's very simple. From what my understanding is, it's better to generally go in the direction of higher resolution to lower resolution. Obviously, big brain is the epitome of the higher resolutions. That's the direction we chose to any template, whether it's FS average, FSLR, ICBM. More recently, I think maybe this Kara Garcia and her group, I've seen it done bi-directionally. So you can go from big brain to FS average and back, and then take the average in an iterative way. But in the case for what is under the hood of BigBrain Warp, which is my registrations, I just do unidirectionally from higher resolution to lower.
That's great. Thanks, Lindsay. No, that's perfect answer. Cool.